Hello, beautiful people. It's nice having you here. I'm so happy to be, and I know you feel the same way. My name is Olayinka Abosedeojo, and I am your lecturer for digestive physiology of monogastric animals. As a way of introduction, digestive physiology in animals is crucial for understanding animal health in order to enhance their performance, revamp their nutrition, and overall well-being. This opportunity or this knowledge affords researchers to have a better understanding of the intricate relationship between diet, gut anatomy, and physiology as it relates with animal health. What are examples of the monogastric domesticated animals? We have rabbits, poultry, swine, and cane rats. Now, the monogastric digestive system is divided into two major parts. We have the receiving region and the digestive region. What did I call it? The receiving region and the digestive region. Now, let's go straight to the receiving region. The receiving region consists of the buccal cavity, that is the whole mouth and the parts within it. We have the pharynx, we have the bill or the beak, as noted in poultry. We have the teeth, the tongue, and the salivary gland. We also have the digestive region known as the stomach and the intestine. The intestines, including the small and large intestine. Now, figure 1 shows pig digestive system. Here, please follow my arrow. We have the buccal cavity, as stated earlier. Now, we have the esophagus, which shows the muscular extension or extended tube that passes the food ingested through peristalsis into the stomach, where it is digested, and we have which goes straight to the intestine, where we have the duodenum, the fore part of the intestine, the jejunum, which is the middle part, and the ileum. And this food, after digestion in the small intestine, it goes straight to the colon, that is the large intestine, and undigested food are passed out through the rectum. Now, figure 2 shows the chicken digestive system. Here we have the beak, which is also known as the bill. We have the esophagus. We have the crop, which is the temporary stomach in the chicken. And then from the crop, the food ingested, partly digested food passes through to the proventriculus, which is the glandular stomach. From the glandular stomach, that is which contains mucus and hydrochloric acid, as well as certain enzymes for digestion of food particles. From here, the food moves to the gizzard, which is known as the muscular stomach. It contains grits that helps to break down the food particles, just the way the teeth breaks down food particles, the food into smaller particles for easier digestion. From the gizzard, it goes straight to the duodenum or the duodenal loop. The duodenal loop is the beginning of the small intestine, and it fully encompasses the pancreas, or we also call it the Aslet or Langerhans. This pancreas also takes part in digestion, as well as it also contains hormones known as the insulin and the glucagon, which helps to regulate the blood sugar. The food from the pancreas, we have the small intestine proper. Here we have the jejunum, and we have the ileum, which is the ending part of the small intestine. This portion I'm pointing at is called ileocecal junction. It is the adjoining junction between the ileum and the cecum. In chickens, we have two, so we call it ceca. Now, this is very germane in chickens because it enhances absorption of fluid and salt, which remains after the digestion of food at the small intestine region. And it also contains protozoans and other microorganisms, which also partake in digestion. From here, we also have the large intestine, and the food, undigested particle or remains of the digestion, passes through the cloaca out as feces. Now, This explains further the chicken gastrointestinal tract. As said earlier, the birds have two stomachs. We have the glandular stomach, which is known as the proventriculus, and it also has the muscular stomach called the gizzard. The proventriculus receives food from the esophagus, and then it also helps to secrete mucus, hydrochloric acid, and pepsinogen, which is an enzyme which breaks down food particles. We also have the gizzard, which receives ingesta from the glandular stomach, and the orifice, which empties into the duodenum, which is the beginning of the small intestine. Now, the cecum majorly functions to receive undigested food materials from the small intestine, and it is also known as the first region of the large intestine. Please, what did I call it? The first region of the large intestine. Now, we talked earlier about the ileocecal junction or the valve, which is the portion that combines the ceca with the ileum. Now, the function of the ceca, as said earlier, it helps to absorb fluid and salt that remains after the completion of the intestinal digestion. It is two in birds, it is one in rabbits, and two in insects. The walls of the cecum composes of thick mucous membrane, which enhances absorption of water and salt. Yes. Figure 3 shows the rabbit digestive system. Yeah, most of these parts are similar because all of them are monogastric animals. Here we have the teeth, we have the esophagus, the pancreas, the cecum, the rectum, the stomach, and the large intestine, as stated earlier. Now, figure 4 shows is a typical example of a monogastric digestive system, which is more extensive. Here we have the gallbladder. The gallbladder produces bile, which helps to emulsify fats. We have the pylorus, which is also like the covering of the stomach, which opens and closes during digestion. We have the duodenum, which we have discussed earlier. We have the pancreas, we have the esophagus, we have the stomach, the jejunum, which we have discussed earlier. We have the ileum, the cecum, the colon, which is the distal, and the proximal, meaning the fore and the hind portion of the large intestine. Now, the parts of the monogastric digestive system, we have the buccal cavity. We have earlier talked about the buccal cavity, and which we'll run through it. Now, it is also known as the mouth. It contains the teeth, the tongue, and the salivary gland. And here, food is chewed, and initial stages of digestion occurs here. Now, the pharynx, also known as the throat, extends behind the epiglottis, which is the cylindrical protrusion found just after at the throat, behind the tongue. It serves as a passageway for both the air and the food. Now, the bills, also known as the beak, is found in birds as well. And the teeth are also part of the digestive system, which breaks down food, by crushing it. Now, the part of the monogastric digestive system, we have the tongue. The tongue is essential for rolling, also enhances chewing and swallowing. Then the esophagus conducts the bolus by peristaltic movement, from the buccal cavity to the pharynx. And this contains a sac-like expanded section called crop. This crop is noted in the chicken. Now, it acts as a temporary storage of food prior to digestion and also enhancing delayed digestion. Now, the salivary gland secretes saliva, which it helps in lubricating and assisting in swallowing. Now, the saliva contains a mucopolysaccharide known as mucin, and this contains amylase and lipase. The amylase, these two are enzymes. Amylase breaks down starch into sugars, while lipase breaks down fats. Now, we are talking about motility of the alimentary canal. Motility has to do with movement, continuous movement of food through the alimentary canal. Motility is very important because it enhances translation of food along the canal and also enhances final expulsion of the fecal material. Secondly, mechanical treatment by grinding and kneading helps in mixing digestive juices and convert food to a soluble form during this motility period. The third one is it enhances mixing of the content so that there is a continual renewal of material in contact with the absorbing surfaces of the epithelial lining to enhance proper absorption. Now, peristalsis. Peristalsis also occurs, is known as a traveling wave of constriction produced by contraction of circular muscle, which is preceded by a simultaneous contraction of the longitudinal muscle and relaxation of the circular muscle. Peristalsis just acts as a pattern of contraction which enhances gradual movement and of the bolus across the esophagus during or via a peristaltic wave. And this enhances proper mixing, proper digestion, and so on and so forth. Swallowing. Swallowing in vertebrates involves integrated movement of muscles in the tongue, and also pharynx is also partakes in it. Now, this action propels the bolus to the stomach. Now we have gastrointestinal secretions. Here we have the endocrine and exocrine secretions, which we will discuss much later. Now, these secretions are also known as hormones produced by ductless glands. We also call them endocrine glands. And this enhances proper digestion. Now, let's go straight to digestive region. The digestive region of the intestine is divided into stomach and the intestine. The digestive region of the digestive system in monogastric animal is noted or is divided as stomach and the intestine. Now, let's go into it. The stomach. The stomach is very important because it provides initial stage of digestion and requires an acidic environment, majorly the hydrochloric acid. In monogastric animal, remember they have single stomach. This single stomach contracts to mix this content with digestive juices. In birds, we have the muscular gizzard, which contain pebbles or sands, and which helps in grinding seeds and grains, which has been ingested by these birds. Now, the small intestine. The small intestine passes through the pyloric sphincter, which is like a door, literally, or part, a tissue that controls in going in and coming out. Going in of food particles in the intestine. It relaxes during peristaltic movement. And this also squeezes the acidic content into the initial segment of the intestine from the stomach, which enhances continuous digestion and acidic and also enhances alkaline, mixing of the alkaline environment. Now, the small intestine is divided into three distinct parts. What did I say? The small intestine is divided into what parts? Can you mention the three parts? We have the duodenum, yes, the jejunum, yes, and the ileum again. The duodenum, the jejunum, and the ileum. Thank you so much. The duodenum is a short part of the small intestine. It's the beginning of the small intestine. It is very short and it also contains or secretes digestive enzymes and which is carried from the ducts, by the ducts from the liver and the pancreas. The jejunum also undergoes, digestion as well as absorption of digested nutrients, and it's the middle part of the intestine, while the ileum is a more posterior region or section of the intestine. It partakes in absorption of digested nutrients, which has already begun in the duodenum and the jejunum. So we have the significance. Now, we have the cells of the liver produces bile salt, and this bile helps to emulsify fats and also neutralize the acidity introduced into the duodenum from the stomach. Now, the pancreas. The pancreas is very, very important organ in the digestive system because it is an exocrine gland. It produces and releases pancreatic juice. This pancreatic juice now contains enzymes such as proteases, lipase, carbohydrates, which are essential for digestion in animals. Enzymes are usually released in their inactive form, which is called zymogen. What did I call it? Zymogen. Zymogen is the inactive form by which enzymes are initially released. And so this now needs to be activated. For example, trypsin is not produced as trypsin initially. It is produced as trypsinogen. This trypsinogen is a form of zymogen, which is an inactive form of the enzyme. And so, by the time this trypsinogen is now activated, it now becomes trypsin, which is now, takes part in the digestion or breaking down of food substances. Now, the intestine contains large number of bacteria, protozoa, and fungi, which multiply continuously to enhance or to contribute to enzymatic digestion. And they are also digested themselves. Please, I want you to note that. Okay, so uh, the intestinal epithelial anatomy. Now, the intestinal epithelial anatomy is very, very important because this is the core or the crux of this particular class. Now, it is a common, you already know, I trust that you already know that a group of cells grouped together, to perform specialized function forms tissues. A group of tissue that perform particular function is known as organ, while a particular group of organs which takes part in a particular function is known as system. Now, we are referring here to digestive system. Now, under the digestive system, we have a lot of organs that makes the digestive system. Now, the digestive system comprises of small intestine. So, the small intestine is an organ which contains a group of tissues, and the tissues now contains a group of cells. Yes, I hope you understand that. So, let's pick it from small intestine as an organ in the digestive system. Now, the small intestine consists of, as I said earlier, tissues. Now, so the lining of the small intestine consists of columnar absorptive cells, which forms monocellular epithelium. Now, epithelium are coverings of cells. Epithelium are coverings of tissue of part of the body. It could be internal or external. Now, the type known as columnar epithelial cell is cylindrical in nature. It's like a cylinder in shape. Now, based on that, I'll go straight to figure 5, which shows the crypt depth and villus height. Now, the small intestine, studying the small intestine under the microscope will show these protrusions, these finger-like protrusions, which is known as crypt depth. This CT is known as crypt depth and villus height. This villus height can extend up to 1 millimeter in length, and this many villus heights enhances higher or increased surface area for absorption in the small intestine. And the crypt depth is the base. I want you to imagine a mountain and valley. Mountain is protruding, tall there. The base is not like valley, which is circular or oval around the mountain. So I want you to picture that the villus height and the crypt depth is just like a mountain and valley. Okay, so let's go to figure 6. Now, the first figure is expressing the intestine at the cellular level. That is, when we come down to the cellular level of small intestine. Now, I want you to know, and I believe that you remember, that a typical animal cell consists of cell membrane, which is selectively permeable, permitting substances, regulating substances that goes in and out. Then we have the mitochondria, which represents the powerhouse of the cell, which helps to generate energy in form of adenosine triphosphate ATP, through which food particles are broken down to release energy. Then we have the rough endoplasmic reticulum, which helps to transport substances within the cell. Here we have the desmosome. This desmosome and tight junction are similar in function. The only difference is that the desmosome, they are specialized cellular, parts of the small intestine, mm, that provides strong cell-to-cell adhesion, and they particularly enhances connection between cells. While we have the tight junction to also act as a seal between adjacent cells. That is, they play a crucial role in maintaining epithelial and endothelial barrier functions. They help to regulate permeability and tissue polarity. Now, this microvilli, looking at figure B shows absorptive cells. This absorptive cells is also known as enterocytes. What did I call it? Enterocytes. This enterocyte, site means cell. Entero has to do with small intestine. So these absorptive cells are also known as enterocytes, and these enterocytes are the cells of the small intestine. Now, the cells at the cellular level, these cells of the small intestine, they are absorptive cells. Their major function of significance is that they enhances absorption of nutrients. Now, this particular cellular portion of the small intestine also contains goblet cells. What did I call it? Goblet cells. These goblet cells are such specialized cells that produces mucus secretion, and this mucus creates a barrier which prevents microorganisms and foreign particles from having access to the blood, to the internal portion of this uh, small intestine. Now, the cellular portion of the small intestine is highly vascularized. What did I say? It is highly vascularized. That is, it contains a network of veins and arteries. These veins and arteries, as I'm follow the arrow here, these veins and arteries enhances transport of this digested nutrient to all parts of the cells of the body from the small intestine. Now we have the central axis. This is just like the major transporting blood vessel that partakes in the transport of these digested nutrients. Now, this particular figure C, as I'm pointing to, contains a further extension of the cell, the enterocyte. Now, the enterocyte contains microvillus, which is situated or added to it. The enterocytes have microvilli attached to it. So this cell is enterocyte or absorptive cell. On top of this enterocyte or absorptive cell is microvilli. What did I call it? Microvilli. This microvilli enhances absorption. It enhances the absorptive performance of this enterocyte. I said earlier, don't be confused. The enterocyte is also known as absorptive cells. What they do basically is it helps to absorb digested nutrients into the bloodstream, which now passes to all cells of the body. Now, the microvilli is the portion of the small intestine that encourages or enhances or stimulates this absorption to take place in a faster dimension. It's just like catalyst. Catalyst speeds up the rate of chemical reaction. Also, this microvilli helps to stimulate better absorption of the absorptive cells or the enterocyte. Now, this figure C is further showing this microvilli in a broader dimension. So can you see this is the microvilli exactly as it is stated here? Follow my arrow. Here is not just like this particular protrusions in dark color is further extended here. So this is the microvilli. Under the microvilli also we have the actin filaments added. We have the cell membrane. All these ones are situated within the cell membrane as it is. So we also have the myosin filament. We have the terminal web. This terminal web is enhancing the connection of this microvilli on top of the absorptive cell or enterocyte, which is the cell of the small intestine. So these extensions, these filaments, all these actin filaments and so on, the microvilli, all those parts are they are there to increase the surface area of the small intestine to absorb more nutrients. Please, I want you to repeat this after me. These extensions, these microvilli, filaments, and so on and so forth, are there to increase the surface area of the small intestine to absorb more nutrients. Yes, thank you so much. I hope you understand. Now, let's go further. Now, each of these absorptive cells contains the brush border. This, which is also known as this densely arrayed microvilli. They could occur in several thousands per cell. So you are looking at it, but it's in thousands per cell. So it's in order to enhance better absorption. So the interaction is there's a rhythmic motion of this microvilli, which results in enormous elaboration of the absorptive surface area. And this occurs continuously in the cell. So we have different folds. We have the villi, microvilli, which enhances an increase of the surface area to at least 500 times, to a total of 200 to 300 millimeter cube. Yes, that's it. So we have the water absorbing and eliminating region. Now, the small intestine also have this water absorbing and eliminating region. The final portion of the alimentary canal helps to remove excess water from the intestinal content, and this enhances consolidation of undigested material into feces, which is now expelled through the anus. Now, this occurs at the tail end of the or later portion of the small intestine and the large intestine. Now, let's talk briefly about exocrine glands. Now, we have the exocrine glands consist of different mixtures of different molecules, and this consists of water, ions, enzymes, and mucus. And a good example of this exocrine gland is found in the salivary gland, in the secretory cells of the stomach, intestinal epithelium, and that of the liver and pancreas. And these exocrine glands typically consist of invaginated epithelium or closely packed secretory cells lining a blind cavity called acинus. This acinus is a small sac-like cavity in a gland which is surrounded by secretory cells. They are surrounded by secretory cells. Now, these secretory granules are found within these acinus cells, and they are synthesized into granules in the Golgi apparatus. Now, we have different types and forms and methods of secretions. We have the exocytosis. Yes, can you mention it? What are the form, means, and methods of secretion in monogastric animal? Oh yeah, let's go. Exocytosis, apocrine secretion, merocrine secretions, and holocrine secretions. Yes, thank you so much. Now, let's go into the detail. The exocytosis entails the fusion of the membrane surrounding secretory vesicles with the cell membrane such that the contents are expelled from the cell. That is exocytosis, then out of cell. Cytosis means cell, exo means come out or move out. So it is also regulated by the level of intracellular calcium. Now, secondly, the apocrine secretion. This apical portion in apocrine secretion contains the secretory material which is sloughed off and literally removed, and the cell then releases its apex, which occurs just like the top portion is the topmost covering is opened, and then the secretion starts gushing out or flowing out. Now, for merocrine secretions, the apical portion is pinched off, and the containing product now breaks open in the acinus. Now, it is still within this secretory portion that the secretory product breaks out and it starts oozing out. Now, the holocrine secretion, the entire cell is cast off, and just like a breakdown of the total acinal cell in order to release its contents. Now, let's briefly talk about the bile and the bile salts. The bile in the vertebrates, liver does not produce digestive enzyme, but they produce bile, which helps to emulsify fats. It basically contains a mixture of cholesterol, lecithin, inorganic salts, bile salt, and bile pigments. And this is manufactured from the cholesterol. They are also conjugated with amino acid complex with sodium. And this cannot occur without the breakdown of hemoglobin metabolite biliverdin and bilirubin, which are breakdown from red blood cell, mature red blood cell when it's to be broken down. Now we have fermentation. Fermentation is a metabolic process which occurs where microorganisms such as yeast, bacteria, convert carbohydrates, that is starch and sugar, into other substances such as alcohol through an anaerobic metabolism. Now, in monogastric, fermentation occurs in the cecum and in the large intestine, that is, they are hand-gut fermenters. The ruminants are foregut fermenters. Now, microbes in these areas break down undigested carbohydrates and produce volatile fatty acid and other metabolic activities. But this process is very inefficient in ruminants. Uh, yeah, comparing it with ruminants. Now, we have the fore and hindgut fermenters. As I said earlier, foregut fermentation occurs in specialized stomach compartment before the acidic stomach. Ruminants have four stomach compartments. We have the rumen, the reticulum, abomasum, and omasum. So they are foregut fermented. That is, their fermentations occur before it gets to the true stomach. But in monogastrics are hindgut fermenters. They ferment, their fermentation takes place after the true stomach, that is in the cecum or colon. Now, and this food passes through the small intestine, the nutrients are absorbed, the remaining undigested food particles are now fermented in the cecum and in the colon. Yes, digestive enzymes in the GIT, that is gastrointestinal tract, is as shown in this table, summary table. This summary table enhances the show or the expression of different enzymes in different portions of the GIT and the names of those enzymes. Now, at the buccal cavity, we have salivary amylase, which sometimes breaks down carbohydrates. We are in the stomach, we have pepsin, and we have gastric lipase. In the pancreas, we have amylase, we have trypsin, and we have lipase, which breaks down carbohydrate, the protein. The trypsin breaks down protein and lipase breaks down fat. In the liver, we only have bile salt that is produced. That is the only enzyme seemingly produced in the liver is bile. Then in the small intestine, we have the sucrase, the maltase, and lactase, which breaks down carbohydrates. And in the small intestine, we have the endopeptidase, which breaks down protein, and there's no effect, there's no enzyme that breaks down fat in the small intestine. Wow, it has been a very wonderful time. I really appreciate you. Thank you for your time. Thank you so, so much, and see you next time.